All right, hello everybody. It's our first live feed from Grouper Moon 2019. I'm so excited to be here with you guys. I can see all your smiling, happy faces out there. We have a bunch of stuff that we're going to share with you today, but I just want to tell you a little bit about what's been going on so far. Again, my name's Todd. I've been talking to some of your students so far, some of your teachers, but just so you know, I'm an educator in Seattle. I teach middle school language arts and social studies, and I get to do this amazing education project in the Caymans every year. I took 36 hours to get to Little Cayman from where I live. And actually where I live, there was 23 inches of snow on the ground when I left. So this is a nice change of pace. Everyone from the Grouper Moon is just getting here today. This is actually the first year that we're gonna have, our first live feed is happening on the full moon. This is the actual Grouper full moon is tonight, right? And that's pretty exciting. Hopefully some of you saw that huge moon last night. It's a super moon. So I'm here, and all of the scientists are just now arriving to begin their research out on the spawning aggregation site. Today, I'm going to introduce you to Dr. Bryce Simmons, who is one of the main scientists working on the project. And he's going to tell you a little bit about how he became involved in this project, what his role is, and what's been happening so far as the project for this year gets rolling. So I'm going to scoot over and invite Dr. Simmons over. You have all already seen his face on some of the videos that I've shared with you guys already.
Hi, everybody.
I just want to check with Martha. Can you guys hear me okay? Can you give me a thumbs up if you can hear all right? Yeah, okay. So Dr. Simmons, can you first explain what brought you here? How did you get here and what is your role?
Sure. So in terms of getting here specifically, I took about 30 hours just like you did, right? So, we came from Southern California. There's actually scientists from all over the world that participate in our project every year. In fact, this year we have a scientist that's coming all the way from the Galapagos in order to be here to help us collect data. So we have a huge program of different scientists that come from all over the place that all have their own expertise, their own particular skills. And that's really great because it helps our program to be very diverse and to do some really great science. So I got involved in this project about 10 years ago. The program itself has been going on for almost 15 years now. And the program is aimed at better understanding what's going on with spawning aggregations of Nassau grouper here in the Cayman Islands, because the Cayman Islands were particularly concerned with their populations of Nassau grouper, having seen recent crashes in the populations due to overfishing.
And you guys all know from the work that you've done already the importance of an apex predator like the Nassau grouper and how important it is in the food web and the ecosystem here in the Caribbean Reef.
Yeah, and what's interesting is it's not just its importance in the ecosystem, but it's also part of the social fabric of the Caribbean. The species has been part of fisheries in the Caribbean for well over a century. And so it's really important culturally as well as ecologically.
Right. So what is your title on this project?
So I'm one of the lead research scientists for the Grouper Moon program. We have several. We have Dr. Christy Simmons,
who you're going to meet shortly.
Yeah, and she's a scientist with the REEF Environmental Education Foundation, which is a citizen science nonprofit, a marine science nonprofit organization. And we also have Dr. Croy McCoy, who's a research scientist with the Department of the Environment here in the Cayman Islands. What makes this project unique is that we don't just bring a bunch of scientists in from all over the place and descend on the Cayman Islands. The program itself is part of the science of the Cayman Islands Department of the Environment. So they are integral to this program.
It's really a partnership.
Yeah.
Which is a really amazing aspect of this project, I think. So can you tell us, everyone's starting to get here today. Usually when we do these live feeds, the Grouper Moon project is already well underway and everything is happening. What will happen to begin the project? What does it look like when it starts? What will you do?
Yeah, so we've had a few people here, but today is the day, this evening, where we'll have something like 9 different scientists fly in over the course of a couple of hours. Today is the first day we're going out on the aggregation site to do a dive. So this is going to be the first day that we actually get out to the site this month and see whether or not the fish are there. So it's a particularly exciting day for us because we don't know at this point whether or not the fish are all out at that aggregation site ready to spawn, or maybe they're not. And so this is our first opportunity to get out there and see what's going on.
Right. Excellent. So some of you in our audience today know about fish that spawn. You've learned a bit about grouper, and also that that salmon spawn where we are in the United States. We always know when the salmon are going to spawn. Why are we unsure when the grouper are going to spawn? And it seems like when I started working on this project 8 years ago, it was pretty much always within a few days of each other. And now it seems to maybe be a little bit different. Can you just talk a little bit about that?
Sure. So what we do know about Nassau grouper spawning is that they tend to spawn shortly after the full moons in the winter in the central part of the Caribbean where we are. So in that winter, they do that in the winter because the winter is the coldest period of the year for water temperature. So the things that cue these fish to spawn that tell them it's time for spawning is both light from the moon and also the water temperature being cold, which we presume is important for the baby fish that are created at the spawning site.
So you guys think temperature really is key?
Temperature is key.
Okay.
And what's really interesting, because here they spawn in the middle of winter, but if you go all the way up to Bermuda, which is very far north and water temperature is much colder, they actually spawn shortly after the full moons in the summer. So they're targeting a very specific temperature window, not necessarily the coldest period of time,
right,
but it just so happens that temperature window here in the Caymans is the coldest window of time. And I should say that that's actually really important because if we think about climate change or a future warming ocean, they're already spawning at the coldest point in time,
right,
during the year. If that coldest point in time gets warmer and warmer and warmer with climate change, we can only assume that that's bad news for reproduction of Nassau grouper.
Sure. And that could extrapolate to salmon as well, and the temperature of the rivers in the United States affecting the how many of those babies survive and recruit.
Yeah.
So you were here last month. I'm going to share a video in a minute of that, but can you tell us why you were here last month?
Yeah, so getting to your last question, while we know that they do spawn in the winter months and they tend to cue on moons, they tend to show up shortly after full moons. In years when the full moon is late in January, they'll spawn in January. And in years when the full moon is early in February, they're going to spawn in February. This year, the full moon was kind of in the middle of January, in the middle of February. And in those types of years, we just don't know which month they're going to cue on.
This is one of the challenges of field science, that you can't control all the conditions, and sometimes things just don't go your way. And there was a year that we all came and none of the fish came. None of them were there.
Yeah.
And that, that's a challenge, and that's something that scientists that work in the field face all the time.
That's right. Sometimes we get it wrong.
Right.
So we thought it was going to be February this year based on previous year's data, but we couldn't take the chance of missing it. And that's why we sent a team in January as well.
Okay. Now, some of you may have noticed on the blog I posted yesterday, and if you haven't been checking, remember that's your homework. I posted a photo of you talking to a new research assistant on the project. Can you tell us what this is? What's happened? This is new.
Yeah, so this year, we actually brought down a scientist, a young scientist, who's actually specifically responsible for helping us manage all of our equipment and manage all of our data. Over the years, as the program has grown and technology's gotten better, we are generating more and more data.
Sure.
We're generating a lot of video data, we're generating count data, all kinds of different pieces of information, and it's getting harder and harder for us to manage that in the field. So this year we brought down somebody whose role is to help us manage all of our equipment, keep all the cameras charged, as well as make sure that all of that data gets downloaded and stored in the correct and proper way so that we have that data for the future, so that we don't lose it.
And her name is
Sophie.
Okay, so I'm hoping we're going to get Sophie on here so you guys can see her, because that's what it might look like for some of you that are interested in becoming a scientist. That's one of the steps you might take, being an undergraduate or working with an organization like REEF at the DOE. Something else that's really exciting about this year is we got a new toy, a new scientific tool to use on the Grouper Moon project.
It is kind of a toy as well.
And we're really excited to share it with you. Bryce, do you want to introduce it?
Sure. So you guys probably have all heard of an ROV. Can we unmute them? So I wonder whether or not anybody is able to tell me what ROV stands for. No, and we're not.
Hold on.
Oh, we got some hands.
Martha, can you try unmuting you? It's not letting me unmute you. Oh, there we go.
There we go.
Okay. Nope. Hold on.
Wait.
Hi.
There.
Okay.
Okay, now we hear you.
I am Montuesca. I'm a transplant.
You're gonna have to speak up. We can't hear you.
Shout it out.
I remember right. It's something about respiratory. Shoot, I just remembered it. I just forgot it.
That's
Crap,
okay. We'll come back to you.
Yep, Sammy.
One word though.
Who thinks they know what ROV stands for? I see a hand way in the back that's having a.
Sammy.
I
Come here,
thought-
Sammy. Come here.
I think
Hi, Sammy.
Up here.
I think an ROV is a remote operated vehicle.
Oh my goodness. You are the smartest ever. That is absolutely right. It's a
Gold star for you, Sammy.
gold star.
Woo!
Yeah, so it's a remotely operated vehicle. And the reason we call it a remotely operated vehicle is because there's no person inside of it. We operate it using a joystick. You guys probably are all familiar with this. That looks probably pretty familiar if you guys have an Xbox or a PlayStation, right? So this is actually our controller for this. This is an ROV. It is a little robot that we can put underwater and we can drive it. And we can drive it using this. And the reason why you're seeing
You see,
people
you can kind
in
of
the
see
screen.
me there in the
Yeah.
screen.
the reason you're seeing that is because it's got actually a camera in the front. So that's the camera. See that right there? And so when we're driving this thing, we can use our screen here and our joysticks in order to control this underwater. Okay, and it's got a big long tether, and this tether goes to the surface at the boat. Why this is important for us is because it's hard for us to keep doing dives over and over again, going underwater with researchers. And it takes up a lot of effort and time to do that. But with an ROV, we can explore areas of the ocean very, very quickly just by throwing our robot over the water and using our controller to tell it where to go. So we're doing that this year to explore some locations where we think fish might be spawning based on some previous tagging work that we did that showed that they go someplace other than where we expected. Okay. And so the cool thing about this is it allows us to quickly, without getting into the water, use a remotely operated robot to go down and explore different areas of the ocean around the islands.
Okay, that sounds awesome. Like we said, Dr. Simmons was here last month and they tested out the ROV when they were here. And I have a video that I'm going to share with you guys so you can see it in action. So hold on one second while I pull that up for you. In Redmond, Washington, which is interesting. Try doing it from down there.
I'm looking at it.
All right. Okay, and I want to welcome, we have another school here in Key Largo. It's so great that you guys were able to join us. We were just sharing a video using here on the Grouper Moon project called an ROV. This is Dr. Bryce Simmons, who's talking with us. And again, my name is Todd, and I'm the educator here for the Grouper Moon project. Welcome. We're going to continue talking, but at the end of this, we're going to have time for a Q&A. So because you're joining in the middle, you may not understand everything that's going on. So just hold on to your questions to the end and we'll be able to answer everything, okay? I'm also going to put you guys on mute while we're talking. All right. Okay, so we just saw the video. How did that go? What was it like using that for the first time here in the winter?
Right. So it worked pretty well. I will say that I kind of wish I had my son with me on the boat because he's way better at using those controllers for the Xbox than I am. So he probably would have been much better at controlling the ROV than I was. I bet you guys all kind of have a similar experience. You're probably better than your parents at using joysticks.
So for those of you that weren't here, this is actually how you control the ROV with this. It looks like a DS. Is that what they're called? This is what you control it from up above.
So, but it went pretty well, in part because we had very good conditions. It was pretty to have flat conditions where we could go out and put the ROV in the water and navigate it down. And you probably were able to see at the very end there were a couple of grouper. One of the really cool things was that we were able to actually locate in January a spawning location that we thought was there, but we had the ROV to actually find for the first time and verify a tiger grouper.
That's awesome. So like Bryce just said, you noticed in the video, hopefully, that the conditions, the beautiful blue sky, the seas were calm, and that's how, but it's not always like that. In fact, it's often quite different. So I'm going to show you just a quick clip of what it looked like most of the time when Dr. was here working on the project. So hold on one second and we'll watch that video. All right, here we go. And you see Dr. Bryce right there with his jacket on. Woo! All right. All right, we're back. Were you guys able to see that? Were you able to see the boat and all the waves and how, yeah. So when it's like that, what do you guys do?
Well, if we can go out, if it's safe enough for us to go out, we'll go out anyway. So because
Really?
yeah, because we're doing science, right? So we're not out there just to have a great dive. And sometimes the dives, they are beautiful, they're amazing. But we're here to do a job. We're here to go out and collect the information that the Cayman Islands Department of the Environment needs in order to better manage the species. And sometimes that means going out in really rough weather, like you said. But if it gets too much rougher than what you just saw, sometimes we'll just have to cancel the dive because the conditions are so unsafe that we worry about the boat being able to handle the waves that we're in. And we never go out when there's any question of safety of our researchers.
Okay. That's how did you get the ROV? We were just talking about the ROV, and I know that there was a program that you guys that came to you. Can you talk just briefly about what that is?
Sure. Yeah. So
I'll hold it up.
the ROV is from a company called Open ROV. The ROV is called a Trident, and they have a program where they work with nonprofit organizations and academic institutions and government partners in order to provide those ROVs, something like this, for free. And what they expect in return is that we generate education and outreach products that say how we're using the ROV to share the word about conservation and marine science. And this is a partnership with National Geographic.
Right.
So we actually have a blog page with National Geographic that has all kinds of recent videos that we've generated with it, as well as kind of short snippets about the information that we're getting with the ROV.
And I'm going to post the link to the National Geographic blog for the Trident on the Grouper Moon Education blog today, and you guys should go on there and check it out. And we haven't made it official yet, but we were talking last night, and I felt like this is really a new research assistant on the program and doesn't have a name yet. And we were thinking maybe one of our classrooms could help name our ROV. So if you could think of an interesting, cool, appropriate name for our ROV, you could share that with us. That would be pretty awesome. Now, Bryce, we have other really cool tools that you scientists use to study the NASA. I've seen you people out using lasers, and I'm not sure, what are you doing with lasers and the fish? Are you hurting them?
No, I don't think it hurts them. We do use lasers. Let me show you what the setup that we use. This is one of our laser setups. Kind of looks like a space-aged device, right? And each of these gold pieces of equipment is actually a laser. And it shoots a red laser out. And both of these things are perfectly parallel. They're exactly 20 centimeters apart. And because these are perfectly parallel, the two dots, or the two red laser dots that these things create when I shoot something with it, are exactly 25 centimeters apart. And it doesn't matter how far or close to that item I'm shooting I am, it's always exactly that distance apart. And so we've got a GoPro in the middle, and we're recording everything that we're zapping with our lasers, and we're going around and we're zapping all the fish that we can at the aggregation site. And we can later on take our video and put it on a computer, and because we know the distance of those 2 points, we can then figure out what the total length of each of the fish that we shoot is. And so we generate lots and lots of lengths of fish from the aggregation site. The reason we do that.
Well, hold on, let's see. We have just 2 classrooms on right now, so I would love to take maybe a couple of guesses. Why do you think these scientists might be using laser calipers that are parallel, meaning that they're the same distance apart and never cross? How is that going to help them on a project like this? Does anyone have a guess? Yeah, I see. I'm going to unmute our school here in Key Largo. Let's see if you guys have an answer for us. We need you to unmute your microphone. So I can't get you to unmute, but if we could get your teacher to type in an answer into the chat block, or you can email me. Oh, there we go. Now you're live. Hey, guys. So awesome to have you here. So who thinks they have an idea? How might the scientists be using calipers? Okay. So,
I think because it will help you see how long the fish can get from amounts of times.
does anyone else have an idea?
Hi.
It could, so you can tell how many fish it might be able to spawn.
Those are all good guesses. In the front here?
Eric.
And look at the heart and how big the heart is.
Those are all excellent guesses. I think we had one correct answer in there, didn't we?
Yeah, we use the lasers and the video in order to get at the sizes of fish. So let me ask you another question, since we're asking you questions. Are you guys bigger than or smaller than your parents?
Smaller.
Do you expect that sometime in the future you will be about the same size as your parents?
Okay.
So would you say that in the future when you're the same size as your parents, you'll be older?
No.
Yes, you're going to be older when you're the same size as your parents. So we use the size of fish to tell us something about whether or not we're getting new young fish at the aggregation site, right? So if I find a very small fish at the aggregation site, the big moms might be that big, right? But if I find a bunch of these smaller fish at the aggregation site, that tells me that there are new fish showing up at the aggregation site. So it's not just that we go out there and we count how many fish there are. We collect size frequency data or a bunch of lengths of these fish. That also tells us something about how many new fish are showing up at the aggregation site. And we care about that because that tells us whether or not the protections and the management in the Cayman Islands is working.
Okay, so by counting the fish and telling how big they are over the years, you can tell if the fish are getting older and if there's newer ones coming in.
Exactly, yeah. And what's really cool is older fish are important, not just because we know that they've been around for a while, but for instance, older, larger female fish produce many, many more eggs.
Okay, so don't give this one away.
Okay.
We're gonna give this to you guys. Who thinks they know how many eggs an adult female grouper produces when they spawn in the winter? And why don't we go back to Renaissance and get a guess?
Yes! 200.
Okay, good guess. Anyone else have a guess?
Anyone else? Kathleen?
Yeah, 700.
Maybe about 5,000.
Okay, good.
Okay. Ainsley? Last one.
Like 10,000.
Okay, good guess. Let's go on over to Largo and see if they have a guess. All right.
Okay, Adric,
About 50.
50?
What was that?
50?
Oh, who else?
Xander.
Like 50 to 100.
Okay, let's get one more guess.
William.
100.
All right. So, Bryce, how close were they?
Well, it turns out if you were a frog, maybe 50 to 100 would be the right number of eggs. If you were a damselfish, you guys know what a damselfish is? You ever seen how about Finding Nemo? You guys seen Finding Nemo?
Oh, yeah.
Yeah. So there you're talking maybe 5,000, maybe even 10,000 eggs. With a big female Nassau grouper, female Nassau grouper,
Right.
maybe 2 million, maybe even 3.
Whoa.
Millions and millions. And so when we have 3,000 big females at that aggregation site, we're talking billions and billions of eggs that are being spilled out every winter at that spawning aggregation site. So it's a really important location for generating new baby grouper, not just in the Cayman Islands, but everywhere in the Caribbean.
Wow, that's amazing. So there's one more tool that we use that I want to share with you, and then I'm going to introduce you guys to another scientist that's working on the project. Bryce, what's this funny thing?
Yeah,
I don't know
that
if you can see this. This is,
looks a little bit like spaghetti, right?
yeah.
That's actually called a Floy tag. It's a visual tag that we use. We implant that in the back of a small number of fish at the aggregation site using a small modified pole spear. And we go at the aggregation site, and we go down and we implant about anywhere from 50 to 100 of these tags in the thousands and thousands of fish that are there. And once they're in the back of the fish, we can see that very easily. And they only last about a year, if that, on the fish. So they're not very impactful. It's kind of like getting an earring or something like that. But they're very useful to us because once we know how many of these tags have been put out, we can then go and we can do counts of tagged to untagged fish over time. So we count, let's say, 100 fish, and 20 of those fish, on average, are tagged. Well, that means we've tagged 20% of the population. Oh, right? Yeah.
So I bet a lot of you are working on percentages this year. Yeah, you know about this.
Yeah. So if we know that we've tagged 20% of the population, and we've tagged 100 fish, then let's say we've tagged 100 fish. Then how how many would we have? How many total fish would we have?
Okay, so let's see if we can get some guesses from the classroom.
I'm going to make it easier for you. Okay. We tagged 10% of the population. Okay, so we estimate that of all of our fish, 10% of them are tagged, and we tagged 100 fish. Okay, how big is the population?
All right, let's see if we can get any guesses. We'll go back over to Renaissance. Does anyone think? Oh, I see some hands up.
I'll bring up.
Come here.
See?
Yep.
Where am I?
Right there.
Where am I?
How much? 10% of 100.
1,000? No.
Yes, you're right. If 100 fish represents 10% of our population, then our population is 1,000 fish. So it's a little bit of math, but it's not all that complicated, right? But it's a very powerful tool. All we need to do is be able to come up with a good estimate of what percent of the population we've tagged.
Now, so I've been on this project for quite a few times, and when I count something in class, when we're counting something, we usually just count it once, maybe twice to be sure. But here on the project, you guys are counting 3 times a day with multiple people out there counting. Why do you have to count them every day? Why?
Well, at the end of a week, we'll get hundreds and hundreds and hundreds of counts we count 50 fish, and we keep track of, every time we count 50 fish, we keep track of how many of those are tagged. And we do that over and over and over again. The reason is because we tag such a small proportion of the population, because we don't want to have an impact on the population. So we tag as few as we can get away with. But what it means is when you have a very small portion of the population tagged, in any given count of 50, you'll often get 0 tags. And so if we're trying to estimate a very small proportion from that data, we need to do counts over and over and over again to come up with an accurate estimate of the proportion of the fish that are tagged, or that percent.
Wow. So you guys, I'm going to be posting some more information about these tags and about how the scientists count them and how that works on the Grouper blog. And like I said, I'm going to be posting on the Grouper blog every day, cool behind the scenes videos, interviews, photos and stuff. So go on there every day and check it out. And you can leave comments and I'll respond to you that same day. It's super fun. Bryce, can you say goodbye? And everyone, can you say thank you to Bryce for being here?
Bye, friends.
And I'm going to introduce you to another scientist that's here. All right. Welcome.
Todd.
Okay, so this is Dr. Christy Simmons Pattengill. Can you tell us, okay, first, how did you become Dr. Simmons Pattengill and what is your role on this project?
Well, I am the Director of Science at REEF Environmental Education Foundation, this organization that Bryce talked to you about. And long ago, I did my PhD research on fish. I studied fish.
Where?
Fish at a place called the Flower Garden Banks in the Gulf of Mexico, which is a national marine sanctuary. It's a very special place. You wouldn't expect to have coral reefs out in the Gulf of Mexico, but there they are. And I got to do that. But before I even made it to graduate school, I was just in college, and I spent a summer in Key Largo, and that was right when REEF was starting. And so that's how I got involved in REEF. And then when REEF started the Grouper Moon Project in about 2001, we started to get involved in figuring out what science we were going to do to help understand and save the Nassau grouper.
So Dr. Bryce works at a place called Scripps Oceanic Institute as a professor. You work for REEF. What is your role? What's your title?
Yeah, my title is Director of Science. And so
you're the head of science for REEF.
Yes.
Wow.
Yeah. And in addition to the Grouper Moon Project, which REEF does, most of what we do is called the Volunteer Fish Survey Project, which is a citizen science program. And some of you maybe have participated in other citizen science projects, like doing a stream watch program, or maybe the backyard bird count, which just finished this past weekend. There's all different ways that citizen science programs can happen.
And the idea behind citizen science is that people like you and I that don't have PhDs in marine biology can still help scientists and help understand and add to the body of knowledge that we have.
Exactly. So a lot of what REEF does is help bring in divers and snorkelers and others to help collect information about the ocean.
That's so cool.
Yeah.
So, Dr. Pattengill, Dr. Simmons Pattengill, today is the first day of real REEF project. We've gone out on the dive site yet. So today at 1 o'clock, you're going to go out. We're going to take video, and I'm going to post some of it on the blog, and I'm going to share some of it with you tomorrow. But could you tell us what's going to happen today when you guys go out there?
Yeah. So yesterday we did a dive, but not on the aggregation, over on the regular reefs, just to kind of check out and see if we saw any big Nassau grouper. And we didn't really. We saw several small ones, so that was a good sign. Because
Why is it good? Normally you'd see a lot more.
Yes, normally if we would dive on a regular coral reef around this island of Little Cayman, we would see a lot of big Nassau grouper, but we didn't see any of them. We saw some of the little ones, and those guys are they're too young to go to the spawning aggregation site yet. So probably that means that when we go out there today, we'll see hopefully a lot of big Nassau grouper.
Okay, we're going to ask a question to you guys. Who thinks they can tell us at what age does a Nassau grouper become reproductively mature? At what age can they go? And someone in our class, we'll go here to Key Largo first. Let's unmute you. There we go.
Okay. Lily.
5.
5 years old.
Good guess. Anyone else?
Felix.
1 or 2 years old.
And one more.
How about Zane?
10.
Oh, wow. Those are good guesses.
Those are really good.
Okay.
So what we know from a bunch of science that was done early on a lot actually from the Cayman Islands, and other scientists have told us that the fish are between 8 years old. So the person who guessed 5, that's kind of the youngest that they might go. And then, by 8 years old, they're going. And then we know that they go every year. So they're on their home reefs. They're actually, Nassau grouper are pretty territorial. They don't like other Nassau grouper in their space. They have their own little home spot. They don't move very far from that during the year. But then when it's time to spawn, leading up to that full moon, they all get going, and you'll see rivers of them. They kind of almost are like, if they were talking, they might be able to say, "Oh, come on,
right.
Here we go. It's time to leave." So there's a lot of that behavior that probably clues in as the adult Nassau groupers see others leaving. They know it's time to go, and they all follow each other to this one site on the island.
And so Dr. Simmons just talked about, she used the word behavior. And oftentimes we use that term for people. And we don't always consider that animals have very distinct behaviors. And so if you watched the virtual dive that I sent out earlier, you would have seen Dr. Bryce go down and see a Nassau on its home reef. And they're just by themselves, and they're just hanging out, and they're really mellow, and they're not hanging out with other Nassau. And in fact, if another one comes up, they'll push them away. But then when they come out for this one time of the year to that spot, that aggregation spot, it's like a party. They're all hanging out together. So their behavior changes drastically during that time.
Yes. Yeah. And one of the things that you'll notice when you look at the videos from the spawning site is that they don't all look like a regular Nassau grouper. Regular Nassau groupers are brown, light brown and dark brown, and they have kind of these stripes on them and a big saddle at their tail base and a stripe through the eye. But when you see them at the spawning site, there's some that are really dark, some that look almost white, and you can't see that barring at all, and some that are really black and white. And those colors, those costumes that they're putting on, we think are a lot about because they're such territorial fish normally, that's kind of their visual cue to each other to say, all right, we're all here together, nobody's gonna fight, we're all in this spot.
That's so amazing. So today we're gonna, a bunch of the scientists are gonna go out just after this live feed and start counting fish and seeing what's down there. And we're gonna share all of that with you in these next couple of days. I'm super excited that you guys were all here, and I know that one of our classrooms has to leave. I wanna give you a chance, if you have any questions, to ask us some questions. So we're gonna start over at Renaissance, and we can probably take 2 or 3, and then we'll go back over to Key Largo. Everyone be quiet except for the person that's asking a question.
two questions. One, when you're counting or lasering fish, how do you know you are counting or lasering the same, like 10 fish?
Wow, that's a really good question.
Can you repeat the question just so everyone can hear?
So the question was, when we're down lasering the fish to measure their sizes, how do we know that we're measuring different individuals and we're not going around and just doing the same 10? That's a great question. So there's lots of fish up down there. You'll see when you look at the videos, thousands and thousands.
And they're moving all around.
And they're moving all around. So there is a chance that we could maybe get the same one every once in a while. But the divers in their mind are swimming through, and it's really supposed to be a random, you're not having any, thinking about which one. You just are supposed to, and it's a pretty easy thing to do actually, because you're swimming, you're just pointing it, you leave it on the fish for about 5 seconds, and then you go to the next one. And 5 seconds, and then you go to the next one. And you are spending maybe doing that for 20 minutes or so, 25 minutes while we're down on the bottom. And we usually have a couple divers on the team doing that, and we do that every dive. So probably there's some repeat, but for the most part, we're gonna go do as much as we can swimming through the school to get as many fish as we can.
That is so cool. All right, let's have another question.
Go ahead and sit down, and then I'll have you come back up in a second. So who else has another question, guys? Renaissance. Come here, Nisa.
Hi, Nisa.
Hi. What's the lifespan of one group of moonfish?
Yeah, that's a great question. So we talked about how when they're born until about 5 years old, they're going to just stay on the reef. And in fact, when they're really little, when they first come and settle and become little baby fish, they don't even go out to the reef yet. They like these really shallow with a lot of super hiding holes because they want to stay safe and not get eaten until they're bigger. So when they first come in, they settle in these really rocky inshore areas with a lot of seagrass and little nooks and crannies. And then they move out onto the coral reef and they stay there until they're about 5 or 6. Then they stay there, but they start going to the spawning site each year. The oldest Nassau grouper that I think we know about is in its 30s.
Wow.
But most Nassau grouper are going to live up into its teens and maybe 20s.
Is that old compared to other fish?
Yeah, that's pretty old. There are other fish in the Pacific Northwest and Seattle. There's some rockfish species that live to over 100 years old.
Yes.
So there are fish that can live a really long time. A lot of the smaller reef fish only live a couple years, but that's another reason why we really need to be careful about how many fish are fished,
right?
How many are taken out, because if the fish don't start reproducing until they're 5 years old and they live pretty long, you actually can pretty quickly get rid of all of your fish if you take too many of them.
Right. All right, we can take one more question from Renaissance, and then we're going to go over to Key Largo.
There you go.
Do you research only grouper moon, or do you guys research other fish?
Yeah, that's a great question. So she asked if we only do Nassau grouper as part of the Grouper Moon Project, or we look at other fish too. For the Grouper Moon Project, this place where we go off of Little Cayman, as Bryce said, it's a really special place, and it ends up being used actually for a lot of different species of fish. So there's several different types of grouper that use that same spot to spawn. There's other snappers. We've seen over 20 different species use that site to spawn. So it's a really special place, and it deserves a lot of protection to be able to make sure that those fish can keep living on.
Right.
So yeah, there's, as Bryce mentioned, and in the little clip of the ROV, the fish at the end of that clip is actually a different kind of grouper called a tiger grouper. They're a little smaller than Nassau's, and they have tiger stripes. And they're ones that spawn out at the spawning site as well as a bunch of other places. So that when we took the ROV out last month, we were looking at a different tiger grouper spawning site.
And what's amazing, you guys, about this site, I mean, there's a lot of amazing things about this site. We don't know exactly why, but ecologically, it's very important. Lots of species use it. But the Nassau always use that site. And we know that they don't become reproductively mature until 5 or 6. And so they actually, after 5 or 6 years of never being there except when they were born, which they don't remember, they know how to get back to that site.
Yeah.
Which is kind of amazing, right? All right, so let's go over to Key Largo and we can take a couple of questions if you guys have some. So let's unmute you. And there we go. All right.
How about Xander? Wow.
Why would the NASA groupers spawn 2 million eggs?
Yeah, that's a great question.
Was the question, why did they spawn 2 million eggs? Yeah? Okay,
Yeah.
good question.
Yeah. So as we said, these fish are normally just living on their own during the year, and they're not interacting with any others. And then just this one time a year, around the lunar phase in the winter, they all come together here. And so this is their one and only chance to make sure that there are more NASA grouper for the next generation. So
Are you saying that other fish do it more often? Like they can
yeah,
spawn more often?
there are fish that spawn every day. There's a lot of reef fish that spawn once a month. The grouper really only spawn this one time a year. And then, so they're putting all their eggs in one basket.
Right.
But then also once those little baby NASA grouper, they spend about 6 weeks in the plankton, just drifting around. They're kind of at the whim of the currents. And during that time, a lot of them are eaten by all sorts of filter feeders and, they get eaten.
Sure.
So most of them don't actually survive. So very few actually end up back. And then they're spending 6 weeks out in the plankton. And then they hopefully have ended up back close enough to land where they can settle down and find those little nooks and crannies. So they need to have a lot of chances to make it through the survival.
Great question. All right, let's take another question from Key Largo.
How do you know? How do you know that they're 5 years old when they start to spawn?
Great question.
Yeah. So the question was, how do we know that they don't start reproducing until they're 5 or 6 or 7? So fish have, you know how trees, you can figure out how old a tree is by looking, if they look at the trunk and they can count the rings, because every year that a tree is alive, it lays down another ring in its trunk, right? Well, fish have something like that in their ear. It's a little tiny bone called an otolith. And that ear bone lays down a ring for every year that fish is alive. And so early on, we don't ever harvest any of the fish anymore because this work was done a long time ago. So we don't need to keep doing it. But there were fish, they would take the otolith bone out of the head and look at different sized fish and how many, so how big it was, how old it was. And that can, like Bryce was talking about earlier, you can know something about how old something is by how big it is. And they did that by looking at the ear bones.
That's so cool. Did you guys know about an otolith? I didn't know about the otolith. That was new to me.
Another interesting thing I'll say about otoliths is that in addition to laying down the ring, there's information about what that fish was eating and what kind of habitat that fish was hanging out in. So a lot of times you can learn a lot more about a fish's history, not just how old it is, by that ear bone.
That's so cool. All right, let's take another question.
How about Jaden? No. Guys, I'm trying to give new people chances. Relax. Jaden.
What is the scientific name of a NASA grouper?
Oh, good one. The scientific name of the NASA grouper is Epinephelus striatus. Epinephelus is the genus. And so there's a lot of different grouper that have Epinephelus as its genus. But then the species name is striatus. So that's the full, and usually the way it's written is the Epinephelus, the first word, the genus is capitalized, and then the species name is not capitalized.
Awesome. All right. We have another question.
Hi.
How do you tell where the grouper are gonna settle and where do they usually settle? Like spawn?
Yeah. So the question I think is, how do we know where they're going to settle? Right?
Yeah.
So that we have spent a lot of time as part of the Grouper Moon project, not just looking at that spawning aggregation, but then trying to figure out how many come back here, how many baby grouper come back here. So we've spent a lot of time looking for them. And what we know from our friends and scientists in the Bahamas who do a lot of NASA grouper work, they really like this shallow, like I said, hidey holes. There's lots of little crevices and areas for the baby grouper to hide. And so we've done, and usually it's about, a couple months after spawning that they start to get to those spots. And maybe 6 months from spawning, we'll start to see them. And then they stay in that habitat for about a year before they go out into the regular reef. So maybe we have some pictures and videos of a couple years ago when we were doing some juvenile surveys. We could maybe share those to show you what it looks like.
We can definitely.
They fit in your hand. They're so small.
Yeah, the little baby grouper are amazing. And what's also cool is that the dive shops that are here will often report back to the scientists if they are seeing lots of juveniles out there and babies. And there was this one year where there were a ton, and we got to go out and try to catch some and look at them and measure them. And I'll share some of that video with you guys on the blog. We can take another question.
Felix.
How many predators are there? Like, what type of predators?
Oh, yeah.
So what type of predators for the Nassau grouper? Yeah?
Yeah. By the time they get to be adults, not much. Sharks, for sure. And we do see out at the spawning site, sometimes we'll see sharks out there because it's a good, easy spot to catch some fish because there's so many of them. That's about it for the big groupers.
Sharks and people.
Yes.
People are the major predators.
Yes. So a lot of people, as Bryce said, it's a really important cultural food fish. And unfortunately, because they have this habit of all getting together in one spot and at a very easy, you know on the calendar when they're going to be there, that allowed them to get pretty heavily fished, which is why now it's really important that a lot of the protections are just on the spawning site so that they can be there and be protected from fishing. But then that way there's enough for people to fish for the rest of the other time of the year.
Right. So the whole idea is not to stop people from fishing. The idea is to protect your resource so that it can continue into perpetuity, so that your children, your children's children will still be able to go out and see Nassau grouper on the reef. And if we are taking them on the grouper hole when they're spawning, we're not actually allowing that spawning to happen. So that's why we're really protecting them there.
Yeah. When they're babies, when they're in the plankton, they get eaten by all sorts of stuff. Filter feeders, other bigger plankton will eat them. We could actually maybe share. We have some underwater microscope pictures of egg predators.
Do you guys want to see that? I'll post some of that.
Yeah, those
That's
are pretty
cool.
fun. Because they're microscopic, but they've got big fangs because they're eating the eggs.
That's so neat.
And then once they settle and they're in the nearshore areas, little barracudas and all sorts of other things would eat them potentially before they get big enough to defend themselves. That juvenile habitat for them is this nearshore area. You guys in Florida, you've probably heard of mangroves and how important mangroves and seagrasses are. Those are really important baby fish habitat, which is why there's always talk about trying to protect and not get rid of the mangroves and the seagrass habitat, because those are places where those little baby fish need to go first before they go out on the reef.
You can think about those places like a nursery. They're really like a nursery for a lot of the baby species, and it's where they can hide and eat and not be eaten. Let's take one more question.
One more, Eric.
Okay, so, but how can they die out if there's so many?
Great question.
Yeah, that's a good question. So how can they die out if there's so many? So we're really lucky here in the Cayman Islands, because this spawning aggregation was found, discovered by some fishermen in about 2001. And in a very short amount of time, because they're so easy to fish when they're all together in one place, because remember, they're normally territorial and spread out. But it's almost like they were all put into a bucket, and then it's just easy to remove a lot of them. So in the first couple of years, there were about 2,000 fish that were taken out of just during the 10-day spawning season. And so fortunately, the Cayman government realized that if they wanted to save this population, they needed to close the spawning site to fishing so that we could better understand and study what was going on. And the work that we've done over the last 10 years has kind of shown us that it's probably the best idea to not fish the fish when they're at the spawning vegetation. But when they're, so here, because they've been protected, the spawning site's been protected for so long now, there are a lot of fish. There's about 7,000 fish, maybe 5,000 to 7,000 fish that come here. But in a lot of places, that protection didn't happen soon enough. And so it's taken a really long time in places like Florida and the Bahamas and Bermuda to let the mastitis grouper populations build back up. So interestingly, because they're spending time that first 8 weeks or so in the plankton, 6 to 8 weeks, there is an opportunity for this really healthy population to start to see some other locations. So that's the hope, is that
So if you guys think about it, like in the United States, sport hunters can't go out and hunt in the spring. Do you guys know why?
No. Why? That's just spawning.
Does anyone have a guess why you can't go out and hunt the animals in the spring?
Tegan.
Because maybe so that they can reproduce.
Exactly. So in the springtime, those animals are reproducing. We don't want to hunt them before they've had their babies, because then we're really going to lose the population. We want to allow them to have their babies. Does that make sense? So it's the same idea with this spawning site. During that time when they're making their babies, we want to protect them so that we can be sure that we have new young grouper joining the population every year. Yeah? All right. I want to thank you guys so much for being here. Will you guys say thank you to Dr. Simmons here? Thank you for being here. Okay, I'm going to wrap up with you guys real quick. Hey, do you guys have a couple of minutes? Teacher?
We don't.
We have one.
So, when I started this, I was showing a video of an ROV that we're using. I'll post that video on my blog, and your teacher should have that blog, and you can go on and check it. And I'll put the video on there, and you can see how the ROV works. And then tomorrow when we're back on, we'll show you tons of footage and show you the fish and all that stuff. And we're so excited that you guys were able to join us today. Was it fun?
Yes.
Awesome. All right. Well, thank you so much, you guys. We'll see you tomorrow, okay?
Thank you,
Thank
so much.
Bye-bye. Bye-bye.
Bye-bye.
Bye-bye.